Welcome to the lecture series in food biotechnology. In this lecture series, we are dealing with how the various technological advances and the processes which have come up in the field of biotechnology, how they are useful and how they can be applied to the field of food technology, to the processing of the food, for the preservation and the various aspects. This is part 4 of this particular series where we have been dealing with fermentation, its applications, its nutritional aspects and the health benefits. From time immemorial, fermented foods have been a part of the human diet, and with the advent of science, people have known about the various microbes which ferment the food, and with the advances in microbiology, which cultures can be used for fermentation, which particular cultures give a particular flavor or an aroma or a specific taste to the food, and each nook and corner of the world has its own specific fermented food which is supposed to be the local delicacy or the local food, the staple food or the standard food which is specific to that particular area. So, when we talk about microbes and food fermentation, as you know, microbes can also cause food poisoning, they can also cause foodborne diseases. So, what are the microbes which we use for this fermentation, and generally what is the safety of this fermented food? Because remember, some of the microbial metabolites can also cause food poisoning. It is not the presence of the microbes alone, but some of the metabolites of the microbes can also cause food poisoning, or even it can cause disflavor or not a applicable aroma to the food concerned. So, the first question we are going to be talking about is the safety. So, when we talk about food substrates which are fermented with desirable organisms, see, the point happens is that when a food is already colonized with a microbe, the presence of that microbe can actually prevent invasion or further inhabitation of the food by food poisoning or toxic microorganisms. A typical example of this is the Indonesian tempeh. In this case, the substrate is soaked, partially cooked soybean. This is a specific fermented food. The substrate is soybean, and during initial cooking and soaking, soybean undergoes fermentation. Acid fermentation is what happens here. So, this acid fermentation actually lowers the pH of the food. So, when it becomes an acidic pH, many microorganisms cannot colonize that particular food, but fungi can colonize, they can grow in the acidic environment. So, this fermented soybean now gets cooked and they get dried, the surface of the food is dried. So, when the moisture content or the water activity lessens, the spoilage bacteria will not be able to grow because they require a specific moisture. The organism which grows here is rhizopus. So, this rhizopus now knits the cotyledons into a cake-like substance, a compact cake-like substance. This can be stored, this can be sliced, and this can be used as a protein-rich meat substitute because the substrate is a protein-rich soybean. And the rhizopus which grows here uses the available oxygen and produces carbon dioxide, and this carbon dioxide atmosphere is actually inhibitory to many of the spoilage organisms. Hence, this fermented food also has a shelf life. If this fermentation has not happened and the pH of the substrate has not been lowered, that is, if it is unacidified, then food spoilage as well as food poisoning microorganisms can start dwelling, inhibiting the particular soybean. So, artificial acidification is usually done in the case of tempeh production by introducing Lactobacillus plantarum. While soaking the soybean itself, Lactobacillus is added to the soak water. This is taken as an example. The food fermentations which can improve food safety, that is a safety, that is which can prevent the growth of spoilage or toxic microorganisms are food substrates which are rapidly overgrown by edible microorganisms, leading to desirable flavors, aromas, and free of toxins, are resistant to the growth of food spoilage organisms. The fermentations which involve lactic acid production, fermentations which involve ethanol production, fermentations which involve acetic acid fermentation, fermentations which induce highly alkaline conditions with the liberation of ammonia, and food fermentations which involve or which happen in the presence of high salt concentrations. When we talk about food, there are two major problems in the world: starvation in one corner and obesity on the other corner. So, there has to be a balance between these, because when you have more people starving, there you need to make more food available, and to reduce obesity we need to tamper food in that particular aspect. Again, in developing or underdeveloped countries of the world, because nutrition is not sufficient, we have the deficiency diseases like kwashiorkor, marasmus, etc. So, if we can induce or give or supply more fermented food to those areas, the nutritional impact of these fermented foods, especially on these nutritional related diseases, can both be direct as well as indirect. Generally, fermented food or food fermentation, which can increase the protein content or improve the balance of the essential amino acids or improve their availability, this will have a direct curative effect. Meaning, if the food already is nutritious, but some of the components are not bioavailable, or you get a little less amount of protein or an essential amino acid content, if by fermentation of the same food, if we can improve the protein or increase the amount of amino acid or make the food more bioavailable, then it is a direct impact on the nutritional combination of the food concerned. Likewise, fermentations that raise the content or availability of vitamins such as thiamine, riboflavin, niacin or folic acid, they can have direct effects on the health of the consumers. This is especially true of people subsisting largely on maize, where niacin or the nicotinic acid content is limited and pellagra is incipient, and also in people subsisting principally on polished rice, which contains limited amount of thiamine and beriberi is incipient. So, in corners of the world where maize, rice, etc. are the staple carbohydrate source, these staple carbohydrate or the grains do lack certain specific amino acids which has to be supplied via other food content. There is a nutritional deficiency here. If it is an underdeveloped country and the staple diet is maize or for example rice, and there is no other food which can supply the essential like niacin or the vitamin B6 or thiamine, etc. If there is no other food which can compensate for that, then it's that particular pocket or the people who are consuming that, they definitely would be having these nutritional deficiency diseases. So, for this we will have to supply or we can have fermented food which can actually increase the bioavailability of this specific amino acids. So, biological enrichment of food via fermentation can prevent these nutritional deficiencies. A typical example of the biological enrichment by fermentation is one, fermentation with protein. Typical example, the food is Indonesian tape ketan fermentation. Here the staple food is rice. As you know, rice contains starch. This starch gets hydrolyzed into maltose and glucose and then gets fermented into ethyl alcohol. So, what is happening? The rice starch is now fermented. So, the starch solid is lost. So, the protein content increases here. So, the loss of starch solid results in the doubling of the protein content. The second example is bioenrichment with essential amino acids. Again, Indonesian tepe ketan fermentation is the typical example. Here, the microorganisms which ferment will enrich the substrate with lysine, and close to 60% increase in methionine has also been reported in idli fermentation of India, which is again a fermented food. Bioenrichment with vitamins. If the staple food is rice, for example polished white rice, it is deficient in vitamin B1, which is thiamine. So, consumers of white polished rice will have the risk of getting beriberi. This disease is characterized by muscular weakness, polyneuritis, which leads to paralysis and further heart failure. But the microorganisms which are involved in the tepe ketan fermentation, they synthesize thiamine and restore the thiamine to the level which is found in unpolished rice. So, if you eat fermented tepe ketan, then there is no probability of getting a thiamine deficiency, so the disease can be prevented. Tempeh. This is a protein-rich meat substitute in Indonesia, which is made by overgrowing soaked, dehulled, partially cooked soybean. The fungus which is involved is Rhizopus oligosporus or the other related molds. Another example is the Mexican pulque. This is a beverage or the oldest beverage in the American continent. This is actually produced by a fermentation of the juices of the cactus agave. This pulque or the Mexican pulque is rich in thiamine, riboflavin, niacin, pantothenic acid, para-aminobenzoic acid, pyridoxine and biotin. Another traditional alcoholic beverage is the Kaffik beer, which has a pleasantly sour taste and the consistency of a thin gruel. This is a traditional beverage of the Bantu people of the South Africa, and this is generally made from sorghum caphorum, malt and the unmalted kaffir corn meal. Maize or millet can be substituted for the kaffir corn here, and during fermentation thiamine content remains the same, but the riboflavin content of this will double, niacin content doubles, which is very significant, especially for people who chiefly consume maize, because niacin is normally not the quantity of niacin is negligible in maize. So, during fermentation it doubles, which is actually very significant. Palm sap. This is a sweet, clear, colorless liquid containing 10 to 12% fermentable sugar, and this is neutral in reaction, whereas palm wine is a heavy milk white opalescent suspension which contains live yeast and bacteria with a sweet taste and effervescence, and consumed throughout the tropics. This palm wine contains 83 mg per liter ascorbic acid. So, one aspect of the fermented food is bioenrichment with vitamins, essential amino acids, proteins, etc. The second aspect of fermented food is fermentation can reduce the toxin content of the food, especially the soaking and the hydration, and later the defermentation. The raw substrate which is available in the food will undergo modification. Series of changes happen while soaking. So, the whole food product now gets soaked and gets hydrated, right, and then you are going to ferment this. So, the basic carbohydrate, protein, amino acid, lipid content of the food, which is basically what is present in the food, will undergo various modifications during this process. So, these processes can actually destroy the toxins which are present in the food, like the trypsin inhibitor, the phytate and the hemagglutinins which are present in many foods. Even the aflatoxins which are found in peanuts and the cereal grain substrates is reduced during fermentation. Last but not the least, another advantage of fermented food is the reduction of cooking time for this fermented food, especially the lactic acid fermented food. They generally require very little heat in their fermentation and even can be consumed without cooking. Typical example are the pickled vegetables, sauerkraut, etc., which can be consumed as such. Since the traditional as well as the contemporary fermented food require microbes, it now becomes very important to store the required microbes, especially in a pure culture. See, now that we know the microbiology, the flora which is required to produce a fermented food, not only produce, but to have a fermented food with a specific flavor or a specific aroma, then we know this is the sort of microorganism which has to be used. So, which means that we need a pure culture of the particular microbe. It shouldn't be contaminated with, or it shouldn't be mixed with other species or other genera. So, we need to maintain the required flora in a pure state. So, what are the different methods which can be used for preservation? One is cryopreservation, the other one is freeze drying. These are the two methods which has been used to preserve microbes for a long term. But cryopreservation is the most preferred technique basically in most of the biological research centers which store the microbes for a very long time. So, you prepare a culture of the microorganisms. Once the microorganisms grow to a specific level, we will centrifuge them. Okay, so this, when you centrifuge, the media will be in the supernatant, the organisms or the cells will pellet down. So, this now we will mix with carriers or additives and stabilize them, and then this will be preserved, storage, and once stored, till you take it out from the storage level, this can be in that inactive or dormant condition. Once you reactivate, you have to rehydrate and utilize the particular microbe. The other aspect is either drying or by freezing. If it is by drying, we can be talking about freeze drying, spray drying or fluid bed drying. So, different techniques are used in different places for the culture and the preservation, for the preservation of the microbial culture in a pure state. So, let us see the various processes which are involved. The first process is cryopreservation. The advantage is it is a low cost protocol. Cryopreserved microbe stays in a pure form for a long time, and the cell density is very high in these conditions. But the disadvantage is that the storage temperature has to be stable. So, if the storage temperature varies or there is a disruption, then the microbial cells will either get mutated or will undergo some other change. So, that pure culture series will no longer be accepted, and for this cryopreservation you require specific cryoprotectants, chemicals which are cryoprotectants. The second methodology we talk about is freeze drying. This is a very easy technique, it can be easily handled, and the microbes can be stored for a long term. That is the advantage. But you require a freeze dryer, high operating costs are there. Again, you require for a long term storage, you require cryoprotectants. Third process is spray drying. This is a scalable operation where you have a continuous operation and it is cost effective. But again, spray drying cannot be used for all microbes. So, strain dependent storage, spray drying would be effective only for specific strains of microbe, not be effective in other strains. And again, you have to rehydrate the organism to utilize it. So, it depends upon that. The other technique is fluid bed drying. It has a low thermal stress, it undergoes rapid heating, and it is again a scalable operation. Again, this is also strain dependent and rehydration dependent. The other technique which is otherwise used is microencapsulation. This has a very high survival ratio, and the microbes which are encapsulated can be easily resuscitated and utilized, but this is meant for only short term storage, and again it is dependent, the quality of the microbe which is microencapsulated depends upon the storage temperature in which this encapsulated microbe is stored. And again I am repeating, this is a short term storage option, not a long term storage option. So, we need fermented food, we need to know the pure culture, which pure culture of the microbe is the best for producing the particular fermented food. Since we are talking about food, there are certain other factors which also decide which microbe should be used, which is what we call about the rheology of the changes which happen to the substrate when a particular microbe is being used. So, for this, the nature of the raw material before harvest, or if it is an animal, what was the health condition of the animal before it was killed. So, before and then during, once we are doing the fermentation process after introducing the microbe, what are the processes which happen to the particular food, what are the changes which happen to the particular food, and what are the processes which that particular food undergoes during fermentation. That is the mixing, the aeration, the homogenization, then what are the types of enzymatic modifications which happen there, then what is the condition of the fermentation, what is the atmosphere which is present during fermentation, aerobe, anaerobe, facultative, whatever it is, what is the combination which is present, and is the food emulsified. All these will influence the quality of the fermented food, and the fermentative changes will directly affect the basic measurable rheological quality attributes of the food, which include hardness, the texture of the food, consistency, adhesiveness, as well as the viscosity of the food product concerned. Certain microorganisms, especially the lactic acid bacteria, these produce exopolysaccharides. Okay, these exopolysaccharides can be used in food technology or in food processing as thickening agents. So, these EPS or the extracellular polysaccharides, normally they bind to the surface of the cells and they are secreted to the extracellular environment as a suspension or as a mucilage. And these exopolysaccharides generally are regarded as safe. We call them as GRAS, and they actually play a huge role in the formation of the rheological properties of the fermented food. For example, the texture, the stability, the mouth feel of the food, etc. can be influenced by the type of exopolysaccharide which is secreted by the LAB, the lactoacid bacillus. For example, the feel of the yogurt, it is because of the exopolysaccharide, the mucilaginous feel of the yogurt is because of the exopolysaccharide which is synthesized by the LAB. And all the foods will have a specific visually perceived attributes like color, appearance, mouth feel, as well as the overall quality. This is very important. And again, this color can also be influenced by the type of fermentation process, and the color, how do we study which is the most appropriate color, what is the one which most of the consumers accept. So, there are two ways for looking at this. One is by chemical analysis, that is analyzing the pigments which are present, or physically by interaction of the light which is present. And in many food, especially in meat products, presence of the microbes and their metabolites play a very important role in the development of the color along with the other characteristics. A typical example is the dry cured fermented meat products. Typical example, we talk about classification is into whole muscle dry cured meat products like Serrano ham, Teruel ham, etc., and dry cured fermented sausages. So, here the color development as fermentation happens and then the food is dried. The type of microbe which is present actually influences the development of the color here. The dry cured fermented sausages are actually intermediate moisture foods, meaning shelf life is stable, you can keep them in room temperature, and the color which develops in them is a result of the chemical reaction between the compounds which are added by the nitrate, because nitrates are normally added as a preservative, and these nitrates which are added as a preservative will react with the myoglobin which is present in the meat. So, this reaction actually produces bright red colored nitrosyl myoglobin, and this bright red color is actually a very important quality characteristic. If the color is dimmed or if the property is defective, then the consumers will not accept saying this food is not fresh, or I don't want this because it's not attractive enough for me to purchase. So, the use of the starter culture, which is the microbe which is used as a starter culture in industrial DCFS, which is dry curated fermented products, has become very well established in recent times as a means to increase the processing rates as well as to improve the product consistency. So, you require a stable red colored product which consumers will buy. So, for this, the use of the starter culture, maintaining it in a pure form becomes very important. And the microbiota which are used in these starter cultures depend upon the use of nitrate, nitrate or both as the curing agents. The starter culture used for nitrate added fermented sausages contain only lactic acid bacteria as well as staphylococci, whereas for those which are added in nitrate added sausages include lactic acid bacteria, staphylococci as well as micrococci. So, is nitrite added or is nitrate added? That decides the starter culture which is going to be used for the fermentation. Another product which where this also plays a huge role is fish fermentation. This is done both in freshwater as well as marine fish, and this fermentation improves the sensory as well as the hygienic quality of the end product. Here also color development depends upon the process characteristics, but it is also to a large extent influenced by the color of the raw fish fillets, which is what is the type of the fish which you are trying to process. The other fish products, fish paste and fish sauce, these are also stable consumer preferred products because of their salty cheesy like flavor as well as having a aroma. This is again a natural product of the natural fermentation process in the presence of 20 to 25% salt under ambient conditions. Fermented fish is an excellent source of protein which is consumed widely throughout Southeast Asia. The other major fermented food products which are consumed are the dairy products. Here, color of the dairy product is the first sensory characteristic which is perceived by the consumers, and also this will tend to modify other perceptions such as flavor as well as aroma. Milk whey, this is a source of riboflavin, and the presence of riboflavin here provides a yellow greenish color. Milk fats contain carotenoids that gives the slight yellowish color to the milk. The white color of the milk which you see is actually made due to the scattering of the visible light by the fat globules which is present and the casein micellase which is present. And these casein micellase actually disperse the blue light better than the red light, and so skimmed light milk has a characteristic bluish color which is perceived by the consumers. So, in general, when we talk about the color changes in fermented food, depending upon the food type, this color change can be caused by several mechanisms. In fermented meat products, the chemical reaction which is happening between the compounds derived from the added nitrate or the nitrate and the myoglobin, this is the main factor for influencing the color development, along with the meat structural matrix, the composition of the product. The composition of the product also involves the fat content, whether you are going to add spices, then the microbial metabolism that adds to the decline of the pH during fermentation, and the coagulation of the muscular proteins and the processing conditions. So, all these will influence the color development. In fermented fish products, the color development depends upon the redox state of the myoglobin, the lipid oxidation, the natural pigments which are present in the fish, and the processing conditions such as salting, then the happening of the Maillard reaction and so on. In fermented dairy products, most of this color changes are given by the gelation mechanism of the milk. This can be acidic or combined acidic enzymatic. So, when this happens, there is a structural modification of the milk, pH will drop because of the microbial growth, the milk proteins will reach the isoelectric point and form a gel. when cheeses are formed, the specific microbiota which are used will give the characteristic specific colors to the cheese product. And in fermented vegetables, especially by olives, etc., cultivar, the ripening stage, harvesting, salting, pH changes, the composition, the alkalinization, all this will lead to the pigments composition and actually will lead to the changes which happens during fermentation. The bioactivity and the nutritional value of food can also be enhanced by alterations of the food matrix rather than the effective molecules themselves. For example, cooking will loosen the food structure and increase the accessibility or the bioavailability of the minerals and the vitamins or provitamins. Fermentation induces different types of bioactivities caused by hydrolysis and further metabolism of carbohydrates and proteins, biosynthesis of vitamins, structural changes, the release of antioxidants and other compounds with potential bioactivities. Thus, food fermentation is an important means by which diversity can be introduced to the food available all over the world, with also increasing the bioavailability of the vitamin, protein or the amino acid content, giving increased nutritional as well as health benefit to the consumers all over the world. Thank you.